The title of our experiment is the Soil Characterization Protocol by Lauren Vickrey and David Vaudrin.
The problem of our focus is how soil is characterized for the use of construction of buildings and use for vegetation.
Our hypothesis is that the deeper you dig into the earth, soil becomes more compact due to the gravity and weathering. The greater the depth from the surface, the more nutritious the soil becomes, as well as the darker in color.
The independent variable of our project is based where you are digging. Make sure you are digging in a forested location to ensure that you are using local soil samples. We are also using vinegar during the experiment, which is our dependent variable. You must use white vinegar to properly analyze the free carbonates. This will be discussed further in later.
First, let's discuss safety. A forested location means you need to be aware of snakes, spiders, raccoons, and any other ground-dwelling animals. Also, your dig site should be away from trees, not only to avoid roots, but also to avoid killing the nearby trees. If for any reason you do find roots while digging, dig around it. Second, use gloves. They protect you from splinters, blisters, and minor cuts. And third, read the procedures prior to doing them.
The hole will have to be at least 2 cubic feet to ensure leverage while digging. The soil sample depths should be gathered from 0 centimeters, 20 centimeters, 40 centimeters, and at least 75 centimeters. Make sure the sun shines on the profile if possible. Use a trowel to scrape a few centimeters of soil off of the profile to expose a fresh soil face. Determine whether the soil profile is moist, wet, or dry. If the soil profile is dry, moisten it with a spray bottle. Start at the top of the profile and observe the characteristics of the soil moving towards the bottom of the profile. Look carefully at the soil profile to distinguish characteristics such as color, texture, shape, roots, rocks, dark notches, worms, small animals, insects, and worm canals. These observations will help to define the horizons. Working in a straight vertical line, place a marker, such as a golf tee or nail, at the top and bottom of each horizon to identify clearly. Be sure there is a consensus from all the students regarding the depths of the soil horizons. Measure the top and bottom depths of each horizon, beginning at the top surface of the profile. Start with the meter stick or tape measure at 0 centimeters at the top of the profile. Note the depths at each of the horizons and at the end. Record the top and bottom depth of each horizon of the soil horizon definition page.
When measuring the structure, use a trowel or other digging device to remove a sample of soil from the horizon being studied. Hold the sample gently in your hand and look closely at the soil to examine its structure. Come to a consensus with other students in the group on the type of soil structure of the horizon. There are some possible choices of the structure. One example is granular, which resembles cookie crumbs and is usually less than 0.5 centimeters in diameter. This is commonly found in surface horizons where roots have been growing. The next is blocky, which is irregular blocks that are usually 1.5 to 5 centimeters in diameter. The next is prismatic, which is vertical columns of soil that might be a number of centimeters long, usually found in lower horizons. Next is columnar, which is vertical columns of soil that have a white round salt cap on the top. These are found in soils of arid climates. Next one is platy, which are thin flat plates of soil that lie horizontally, usually found in compacted soil. And in certain cases, soil samples may have no structure. These are classified as either single-grained, which is soil that is broken into individual particles that do not stick together. These always accompany loose consistency, and commonly found in sandy soils. And the next one is massive, which is soil that has visible structure, is hard to break, and appears very large clods. Record the structure type in the soil horizon definition page from the site definition sheet.
After verifying the soil type, we will measure the main color and second color. Take a ped from the horizon being studied and note whether it is moist, dry, or wet. If it is dry, moisten it slightly with water from the bottle. Break the ped and hold it next to the color chart. Stand with the sun over your shoulder so the sun shines over the color chart and the soil sample you are examining. Find the color on the color chart that most closely matches the color from the inside surface of the ped. Be sure that all the students agree on the color choice. Record the soil horizon definition page. Record on the soil horizon definition page the symbol of the color on the chart that most closely matches the soil color and covers the largest area of the ped. Sometimes the soil sample may have more than one color. Record a maximum of 2 colors if necessary. Indicate the dominant color and the subdominant color.
Take a ped from the soil horizon being studied. If the soil is very dry, moisten the face of the profile by squirting water on it, and then remove the ped for determining consistency. Holding the ped between your thumb and forefinger, gently squeeze it until it pops or falls apart. Record one of the following categories of the soil ped consistence on the soil horizon definitions page. It is either loose, you have trouble picking out a single ped, and the structure falls apart before you handle it. Note, soils with single grain structure always have loose consistence. Friable, which means the ped breaks with a small amount of pressure. Firm, which means the ped breaks when you apply a larger amount of pressure, and the ped dents your fingers before it breaks. Or extremely firm, which means the ped can't be crushed with your fingers. You need a hammer.
Measuring soil texture. Step 1. Place some soil from the horizon about the size of a small egg in your hand and use a spray mist bottle to moisten the soil. Let the water soak into the soil and then work it between your fingers until it is thoroughly moist. Once the soil is moist, try to form a ball. If the soil forms a ball, go to step 2. If the soil does not form a ball, call it sand. Step 2. Place the ball of the soil between your thumb and index finger and gently push and squeeze it into a ribbon. If it makes a ribbon that is longer than 2.5 centimeters, go to step 3. If the ribbon breaks apart and does not reach 2.5 centimeters, call it. Step 4. Wet a small pinch of soil in your palm and rub it into your forefinger. If the soil feels very gritty every time you squeeze the soil, go to A. Feels very smooth and no gritty feeling, go to B. Feels only a little gritty, go to C. A. Add the word sandy to the initial classification. Soil texture is either sandy clay loam, or sandy loam. Soil texture is complete. B. Add the word silt or silty to the initial classification. Soil texture is either silty clay loam, or silt loam. Soil texture is complete. And C. Leave the original classification. Soil texture is either clay loam, or loam.
When measuring rocks, observe and record if there are none, few, or many rocks or rock fragments in the horizon. A rock or rock fragment is defined as being larger than 2 millimeters in size. Record your observation on the soil horizon definitions page. When measuring free carbonates, set aside a portion of the exposed soil to use for the free carbonates test. Make sure not to touch it with your bare hands. Open the acid bottle and squirt vinegar on the soil particles, starting from the bottom of the profile and moving up. Be sure to use caution and point the bottle directly at the soil, not toward other subjects, especially towards eyes. If vinegar gets in your eyes, rinse with water for 15 minutes. Look carefully for the presence of effervescence. The more carbonates that are present, the more bubbles, which are effervescence. For each horizon, record on the soil horizons definition page one of the following results from the free carbonate test. None, if you observe no reaction. That means the soil has no free carbonates presence. Slight, if you observe very slight bubbling action. This indicates the presence of some carbonates. Or strong, if there is a strong reaction, meaning that there are many or large bubbles. This indicates that many carbonates are present.
Based on our observations, soil retains water at greater depths. Dig out a large soil sample from each soil horizon. Avoid the areas of soil that were tested with vinegar and avoid touching the soil samples so that the pH measurements will not be contaminated by acids on your hands. Place each sample in a bag or other soil containers. Label each bag with the site name, horizon name, and top and bottom soil depths. Bring these samples from the field into the classroom or laboratory. Spread the samples on separate paper plates and sheets of paper to dry in the air. You can place the soil near a window where it will receive light and sun to make the drying process faster. Put on latex gloves so the acids on your skin do not contaminate the soil pH measurement. Put the number 10 sieve on the top of the clean sheets of paper and pour the dry soil sample onto the sieve. Carefully push the dried soil material through the mesh into the paper. Do not force the soil through the sieve or you may bend the wire meshing opening. Rocks will not pass through the mesh and will stay on top of the sieve. Remove the rocks from the sieve and discard. If no sieve is available, carefully remove the rocks and debris by hand. Transfer the rock-free, dry soil from the paper underneath the sieve into the new, clean, dry plastic bags or containers. Seal the containers and label them the same way that they were labeled on the field. Horizon name, top and bottom, horizon depth, site name, site location. This is the soil that will be used for the lab analysis. Store these samples in a safe, dry place until they are used.
After analysis of our data, we have found that the effervescence released on the profile was not recognizable due to the rich construction of the clay. The National Gardening Association verified that clay is the richest in nutrients due to its retention of water. Upon testing the effervescence, we saw the bubbles were very large at the freshly upturned soil clay mixture at the bottom hole in the earth. Due to the ongoing climate of San Antonio, the soil is often dry due to drought. Our city averages 10 inches on an annual basis. The United States Geological Survey classified that the soil content of silty clay decomposition residuum consists of more than 50% clay. The restrictive nature of clay leads to desiccation deeper than 80 centimeters in the ground. This would cause bowing and cracking of construction foundations if they are not properly weighted.
During our dig, we found the roots remained few in number, however they grew in size the greater in depth we went. There were numerous roots from local vegetation closer to the surface. After reaching the depth of 20 to 70 centimeters, the roots decreased dramatically to few yet larger roots. After reaching 70 centimeters, there were no visible roots. We determined that the soil bed from 0 to 4 centimeters was friable, and all deeper samples were firm to break. All soil samples are granular, with no gritty feeling. After measuring each level structure, we verified that 4 centimeters and below were mostly silty clay loam, because it formed at least 2 centimeters in length when wet without breaking. The top surface of 0 to 4 centimeters was silty clay because it formed at least 5 centimeters in length when wet without breaking.